Good morning, my dear students. Uh, I am Dr. Vipin Pathak. Today, I will tell you about the cell communication because this is the social contact of the cells, and this lecture is only for the educational lecture, and this is very helpful for your competitive exams such as the CSIR, ICMR, ERSNet, and GATE exams, as well as your four course also. So, we will talk about the different points. You can take a notebook and as well as the pen because it must be for understanding to the so you can note down to all these things. Here you will see here the different parts of this one, that is how the connective tissue, the question starts before starting to this one. There are certain questions in our minds that because we are the social animals and we know very well about the communication process, and when we communicate to our friends, then our friends will realize to our understanding and whatever we are thinking for him and whatever he is either he or she is thinking for us. So in the same way, our cellular organization also talk to each other, and these cells also talk to. Sometimes these cells talk to other cells, to their neighboring cells. Sometimes these cells talk to their extracellular matrix part also. But the question is how they talk and what are the processes and by which process they can talk and what are the criteria they are following. In the before you are well known about the structure of the plasma membrane as well as you are also well known about the G protein couple receptors in the cell signaling chapters that I explained in your previous lectures. So I am coming here regarding to this one is special context of the cells. So here you will see here there are some apparatus. There are some apparatus and these with the help of these apparatus these cells talk to each other. These cells to talk to each other. If you see here, in case of this one, if you see here, in this case, you will find out here is some is the epithelial part. These are the epithelial cells, and you will see here these are the epithelial cells. And in the same way, you will see here the important points we will discuss. These are the cytoskeleton parts. Maybe the cytoskeleton. If you talk about the cytoskeleton, so as you are well known about that, the cytoskeleton provides the architecture to the cells, and cytoskeleton has three components: microtubules, which are made up of tubulin, and microfilaments, which are made up of actin, and the intermediate filaments. So now, students, the here you will see these are the mechanical. These are the part of the cytoskeleton parts. The mechanical stress are transmitted from cell to cell by cytoskeletal filaments anchored to cell matrix and cell to adhesion. So now the mechanical stress, that means in another point is here that the mechanical stress is also transmitted by the cytoskeleton elements. You remember it, the mechanical stress, whatever the mechanical stress is coming inside to the cells, and now the cells distribute to its stress to the others. इसका मतलब क्या है? इसका मतलब ये है कि किसी सेल कोई भी सेल if any cells comes under the stress process, so then it distribute to its stress to the others. This also provides the information also, us also that never take the stress and just distribute your stress to the others also, right? Okay, as well as the cytoskeletal elements also makes a contact to the cells to the extracellular parts, extracellular matrix, as well as the cell to cell. Now you will see here these are the some adhesive molecules which are present in the in the cell membrane structures of these cells, and these are the junctions. These are the junctions, and through to the junctions, the information is transmits from one cell to the another cell and another cell to the other cells. This is a part of connective tissues, and mostly you are well known about that. In case of the animal cells, the cell wall is not present, but the our connective tissues, the extracellular matrix part, in your extracellular matrix part, some set of proteins are found, such as the collagen fibers. These are collagen. The important question is, it is the most abundant protein in our body. You have to note down this one. It is the most abundant protein in our body, And in this below to this epithelial cells, you are seeing here, it is a basal lamina. It is made up of a basement membrane, and to this below to this basement membrane, here is the part of this is connective tissue. In this connective tissues parts, you are seeing here many muscle fibers. These are the muscle fibers, as well as these are the collagen fibers. Now these extracellular matrix. This is a part of extracellular matrix. Now this extracellular matrix parts contains the fibrous polymers. Now these collagen fibers are the fibrous polymers which are mainly made up of the chondroitin sulfate, and other other biochemicals, as well as you will see here in many other cells also these are collagen fibers and the other proteins also. Now there is a question that these cells have established to its communication in two ways. One way, it established to its communication between cell to cell, that is called the cell to cell interaction, and other way the cell maintains to its its information, transmission of its information to the extracellular parts, that is extracellular matrix parts. So in this lessons we will learn about to all these things. We will learn to about all these things, that how these things are happening. So here you will see here, this is very important, that what are the physical attachments, because here in here here you will see here, this is the epithelial cell contact, and this is the epithelial to non-epithelial cell. You are seeing here, this is epithelial to epithelial cell contact, and that is a part of cell to cell interaction. On the other hand, you are seeing here that this is the epithelial interaction with the non-epithelial part. Basically, it is extracellular matrix parts. So my dear students, there are four main functions distinct the different molecular basis of this one. Number one is we will come to the junction part. There are four types of the junctions. These are the first is the anchoring junctions. These are the first anchoring junctions, and these anchoring junctions, as you are seeing here, these are the anchoring junctions, and these anchoring junctions includes both the cell to cell addition. This you remember it. These anchoring junctions has two parts. One way they interact to one cell to other cell here, and other way they interact with the extracellular matrix part. That means the anchoring junctions. In anchoring junctions, there are two sets. One set is cell to cell interaction, that is basically known as desmosomes, and the other set is the cell to extracellular matrix. You are knowing that the hemidesmosomes. So now, student, both these junction molecules, both the junctional proteins, because these junctions are made up of proteins, and these proteins are anchored by the cytoskeleton fibers. Now, the role of these cytoskeleton fibers is to maintain the architecture of the junctions. तो आपको ये ध्यान देने की बात है यहाँ पर, you should be careful about that. These anchoring junctions, these anchoring junctions makes a contact with the extracellular matrix part, and other hands, these anchoring junctions makes a contact between the cells. Then the second is, we will come to the second part, is the occloiding junctions. The second is the occloiding junctions. Now you will see here, in case of the occloiding junctions, this is one cell and this is another cell. Now these occloiding junctions seals the gap. These occloiding junctions, now you will see here, these occloiding junctions seals the gap between cells in epithelia, and so by at that process, what will be happening, the cell will become either selectively permeable. This cell will become impermeable to the other cells, one hand. On the other hand, this cell becomes selectively permeable. So this is the role of occloiding junctions. Because here is also plays in some important proteins. And the coming sessions we will discuss to all these proteins which are involved in the junctions. So now, students, here you will see here, this makes one cell, they tightly seals the gap. They tightly seals the gap. आपको ऐसे याद रखना है. They tightly seals the gap. By that process, what will be happening? By that process, the cell will become impermeable. Either it become impermeable or it becomes selectively permeable. I think that this is the part of second is occluding junctions. Then we will come to the third is the channel forming junctions. The third is the channel forming junctions. Now here is the some channels are formed between the cells, such as if you remember about these sodium channels, potassium channels, and other channels. So these are the channel proteins. These channel proteins forms some channels between the cells or the transmission of information. Here you are, what we are seeing here. This is a bidirectional transmission of the information through to the channels. And the third last one is the signal relaying junctions. Signal relaying junctions, basically that signal relaying junctions are, this is the structure of the neurons. This is the structure of the neurons. It is a presynaptic neuron, it is a postsynaptic neurons, and some information is inside to these presynaptic neurons. Now these presynaptic neurons send to their information to the postsynaptic neurons. Here you will see here, these are the neurotransmitters. Now these neurotransmitters uh, released to their content in the synapse. Then here is the some receptor proteins are present for these neurotransmitters or these signal molecules or these ligand molecules. Then these ligand molecule or signal molecule bind to bind their receptors. Then what will be happening? A receptor signal complex is formed here. Now this receptor signal complex uh, send the signals from one cell to the another cell. So there are, you remember that there are four types of the junctions. Number one is the again I summarizing to the anchoring junctions. Number second is the occluding junctions. Number three is channel forming junctions, and number fourth one is the signal relaying junctions. Now we'll proceed to the next parts. This is very important, and this is very important for you because definitely it will help you in your competitive exams as well as your MSc. Now you focus on these. Now these are the anchoring junctions. If you remember to the structure of that anchoring junctions, so here you will see here that these anchoring junctions, because these anchoring junctions are uh, uh, supported by these cytoskeleton elements. And here you will see the actin filament attachment sites. This is important. Actin filament attachment site. That means these the microfilaments playing the role microfilaments. Here you will see here, in this case you will see here, the important task is here. This is one to cell junction, that is known as the adherent junction. Cell to cell junction, that is known as the adherent junction. This is the cell to cell junction. This is the adherent junction. And the second part is cell to matrix junction. Cell to matrix junction. This is known as your actin linked cell matrix additions. It is just like as your hemidesmosomes and it is your desmosomes. So now cell to matrix junction, this is actin linked, and this adherent junction, that is desmosome, that desmosomes are also supported by these actin filaments or microfilaments. The second part is the intermediate filament attachment sites. Cell to cell junctions. You will see here. This is the actin filaments, cell adherent junctions, and the actin linked cell matrix additions. But in case of the desmosomes and hemidesmosomes, because the desmosomes are made up of your cadherin proteins and the hemidesmosomes are made up of the your integrin proteins. So now cell to cell junction. These, when the contact is cell to cell junction, that is known as the desmosomes. Cell to cell junction, that is the desmosomes. When the cell matrix junction is hemidesmosomes. When you will say that these are hemidesmosomes and these are desmosomes, these are adherent junction, on the basis of these cytoskeletal elements, on the basis of these cytoskeletal elements, you are saying that these desmosomes and these are hemidesmosomes. If the intermediate filaments supports to these junctions, then you will say that these are the desmosomes. And that junction is between the cell and cell matrix, then it is known as the hemidesmosomes on the basis of morphologically on the basis of the intermediate filament. Now we will come to the next part, that is occluding junctions. Occluding junctions. This is a question. Tight junction only found in vertebrates. This is a question of CSIR. ठीक है, एक बार क्वेश्चन आया हुआ है CSIR के एग्जाम में कि टाइट जंक्शंस केवल वर्टिब्रेट्स में पाए जाते हैं या इनवर्टिब्रेट. तो योर आंसर इज टाइट जंक्शंस आर ओनली फाउंड इन वर्टिब्रेट्स. So now soon tight junctions and septate junctions. These are the examples of occluding junctions. So tight junctions are only found in vertebrates and septate junctions are found in vertebrates. देखिए ये आया है तो ये भी क्वेश्चन बन सकता है. You have to be careful. Now the channel forming junction. Here is the gap junction and plasmodesmata. दोनों क्वेश्चंस आपके एग्जाम्स में पूछे गए हैं CSIR एग्जाम्स में. Gap junction that is found in animals, only it is found in animals, and plasmodesmata found in plants. That means the gap junction is not found in plants. Now the fourth one is signal relaying junctions. The chemical synapse in the nervous system that I explained. The second is the immunological synapse in the immune system. Role of immunobody, antigen interbody interactions, transmembrane ligand receptor, G protein couple receptor. Now the cell signaling contact, delta, notch, afrin, EPH, all are the part of your developmental biology. Anchoring, occluding, channel forming junctions can have signaling functions in addition to their structural roles. Now we will come to the next parts. This is, you will see here, this is a occluding junction, and occluding junction means, if occluding junction means tight junctions and septate junctions. Tight junctions and septate junctions are the examples of your occluding junctions. Here you see the tight junctions. The role of these tight junctions is just to seal the gap, so no transmission of the major molecules between one cell to the another cell. You will see here. The second is the cell-cell anchoring junctions. Cell-cell anchoring junctions. Now you will see here, this is a junctional complex. The adherent junctions connect actin filaments. The adherent junctions connect actin filament bundle in one cell with that in next cell, right? And you will see here, in case of the cell-cell anchoring junctions, in case of cell-cell anchoring junctions, one is the adherent junctions. Here is supported by your actin filaments, and if you will see here, this is also a part of your cell-cell anchoring junctions, desmosomes. Now these desmosomes are supported by these intermediate filaments, and this is supported by actin filaments, right? And this is a junctional complex. The one is the adherent junctions, desmosomes, and this is tight junctions. Now you will see here, these are the gap junctions. Gap junction is an example of channel forming junctions. Gap junctions allow passage of small water soluble molecules. This is very important. केवल छोटे small water soluble molecules. Only small water soluble molecules will transmit through which junction? ऐसे क्वेश्चन आया था, किस जंक्शन से छोटे मॉलिक्यूल्स का ट्रांसमिशन हो सकता है? तो योर आंसर इज गैप जंक्शन, राइट? इन एनिमल्स में वर्ड था, ध्यान दीजिएगा, बिकॉज द गैप जंक्शन इज ओनली फाउंड इन एनिमल्स, इट इज नॉट फाउंड इन प्लांट्स. In case of these gap junctions in plants, that is found plasmodesmata. So now student, all of these small water soluble molecules from cell to cell, एक बार का क्वेश्चन ही नेट का आ चुका है. दूसरा, then we will come to the cell matrix junctions. So this is you will see here, if cell matrix addition anchor, if it is supported by the actin filaments, this is one junction. There are two junctions for the cell to the extracellular matrix interactions. One is your cell matrix addition. This is your hemidesmosomes. This is supported by your intermediate filament, hemidesmosome. And in desmosomes, the protein is clotting protein. We will come to the catenin proteins and integrant proteins also. Now I think that you understand it. If you have any problem regarding to this one, so you can write in the chat section, because someone is writing something. What about the plasmodesmata? Very good, Jagesh, you are very good. Plasmodesmata is a gap junction. Plasmodesmata is just like a gap junction, and they are only found in plants. They are not found in animal cells. You will see here, plasmodesmata are just like the gap junctions. They are only found in plants, and in animals the gap junctions are found. that means plasmodesmata means channel forming junctions. Plasmodesmata are channel forming junctions. I think you understand it. Then we will go to the next part. If you have any problems regarding to this one in understanding and other things, you can write to all these things in your chat sections. I will explain to all these things. Uh, plasmodesmata are the channel forming. These are the uh, channel forming channel forming junction channel forming junction in plants. Okay, we will go to the next part. Now this is very important for you. I close it and it display in my panel if you have any questions regarding to this one. So now we will come to the next parts. This is the anchoring junctions. This is the anchoring junctions because these anchoring junctions are made up of different set of the proteins. This is very important. बहुत important है students. कई बार के एग्जाम्स में ये चीजें पूछी गई हैं, इसीलिए आपको क्या करना है कि बी केयरफुल एंड लर्न टू ऑल दीज थिंग्स, राइट. Number one is the we will explain to all these things. Adherent junction made up of the cadherin molecules. Cadherin is a protein which removes the calcium ions from the extracellular matrix. Cadherin, and I will tell you about the different types of the cadherins also, because the classical cadherins. Here is the word classical cadherin. At first I will explain it, then I will tell you about the classical cadherin molecules also. I will tell you about the classical cadherin molecules, because the role of these molecules is because this cadherin molecules plays a very important role in removing to the calcium ions from the extracellular mediums. So now the classical cadherin. Now in adherent junction, this is a transmembrane adhesion protein, that is the cadherin protein, right? Now then we will go to the N. Extracellular ligand is cadherin neighboring cell and supported by the filaments, actin filaments, and the intracellular anchor proteins, that are the alpha catenin. These are the supported proteins, associative proteins, alpha catenin, beta catenin, and plakoglobin, gamma catenin, and P120 catenin, vinculin, and beta actinin. Now you will see here, in case of the adherent junctions, you will find out all these cadherins, actin filaments, and these intracellular anchor proteins. If anyone they pick up just one protein and ask you the question. Second आते हैं, we will come to the desmosomes. The desmosomes are supported by the cadherin molecules, and here is the two molecules of cadherin. These are named as desmogloin and desmocolin, and the extracellular ligand. Extracellular ligand is desmogloin and desmocolin in neighboring cells and supported by these desmosomes intermediate filament. This is question, because here is this adherent junction is supported by actin filaments. Now desmosomes is supported by intermediate filaments. So in terms of diameter, they can also ask you. So they in intracellular anchor proteins, these are the intracellular anchor proteins: plakoglobin, gamma catenin. Gamma catenin is also here also involved. Now the plakophilin and desmoplakin, these proteins. So rest of these proteins. अब यहाँ पर दो क्वेश्चंस बनते हैं आपके, जैसे गामा कैटेनिन है, गामा कैटेनिन के ये दोनों ऑप्शन दे सकते हैं आपको कि कोई कौन सी ऐसी प्रोटीन है which is act as an intracellular anchor protein in both adherent junction and desmosome. So here is the plakoglobin, that is a gamma catenin. तो याद रखिएगा, इस तरह का क्वेश्चन वो आपसे बना सकते हैं. दूसरा सेल टू मैट्रिक्स इंटरेक्शन, सेल से मैट्रिक्स का इंटरेक्शन. तो नाउ एक्टिन लिंक्ड सेल मैट्रिक्स एडिशन, सेल actin linked cell matrix addition. Transmembrane protein is integrate protein, and this is the extracellular matrix proteins. Extracellular matrix protein, whatever the protein present in extracellular matrix, supported by actin filaments. The proteins anchor intracellular inside to the cell. These proteins, what are these? Talin, vinculin, alpha actinin, and filamin, paxillin, focal adhesion kinase (FAK). And the second is हेमिडेस्मोसोम. इससे क्वेश्चन एक बार आ चुका है नेट में. हेमिडेस्मोसोम प्रोटीन इंटीग्रिन, ठीक है, इंटीग्रिन अल्फा 6 बीटा 4 टाइप 17th कोलेजन बीपी 120, राइट। ये इंटीग्रिन नाम से क्वेश्चन था, सेज भी आ सकता है ये कोलेजन बीपी 120। एक्स्ट्रासेलुलर मैट्रिक्स प्रोटीन्स एक्ट एज ए लिगेंड सिग्नल or supported by intermediate filaments, and this is the lectin and dystonin BP 130. So these ये proteins they ask to these proteins to you. So I will just talk about some regarding to the classical cadherin, the cadherin molecules. Now the students, cadherin is a protein. Cadherin is a protein, and now cadherin is a superfamily of the protein. Just like as integrin also a family superfamily of the proteins, because these all proteins comes under to this cadherin superfamily proteins, and all these proteins comes under to this integrin superfamily proteins. So what is cadherin? So cadherin is a superfamily protein because it contains many proteins inside to this one, and you will see here the and this cadherin protein. Now this cadherin protein is found. You have to note down to all these things in your notebook also, which is present in your table. Cadherin is present in all multicellular animals. Cadherin is present in the genome of all multicellular animals. सारे multicellular animals के genome में ये present है, जिनका genome की mapping हो चुकी है, ठीक है। Now even then it is also found in choanoflagellates. Even this cadherin molecule is also found in choanoflagellates, and but in fungus and plants, cadherin molecules are not present. In fungus and plants, you have to note down this. In plants and fungus, cadherin molecule is not present. And in bacteria and archaebacteria, there are two groups of the bacteria and archaebacteria. When eubacteria and archaebacteria, they are cadherin molecules. Cadherin protein is not found in archaebacteria and eubacteria also. Basically, this cadherin protein is it is found in animals. Basically, this cadherin protein is found basically in animals. You have to note down this one. The cadherin protein is basically found in animals. The cadherin means it is calcium dependent protein. Cadherin indicates that cadherin is a calcium dependent protein, because this protein removes the calcium from the extracellular matrix. The question is how it removes. So because it has some calcium binding sites, it has basically approximately 3 calcium binding sites, 3 calcium molecules bound to these cadherin molecules, and these cadherin molecules removes the calcium ions from the extracellular matrix. You have to note down these things. These are the important things for you for your examination point of view. Now if cadherin molecule is bounded with calcium ions, now it becomes a complex of calcium cadherin complex. So for removing to that calcium, we are using an enzyme that is known as trypsin. You have note down it. It is a question for your competitive exams, definitely. Trypsin molecule. We are using some proteases such as trypsin or removing to that calcium from the cadherin molecule. And these cadherin molecules, it is a classical and monoclassical cadherins. Classical cadherins and monoclassical cadherins. So the cadherin molecules, these are the classical cadherins. You will see here the different types of the cadherin molecules. The classical cadherins and the classical cadherin molecule, these are the transmembrane proteins. This is the classical cadherin molecule. It is E-classical. It is known as आपका E-cadherin. Because why it is called E-cadherin? Because it is present in epithelial cells. This cadherin molecule is present in epithelial cells. So therefore, this cadherin is known as the E-cadherin. This cadherin molecule present in epithelial cells. So therefore, it is known as E-cadherin. This is a fat-like cadherin. Here are some domains. Fat-like cadherins. These are present in fat cells, especially in fat cells. So now E cadherins is present. E cadherin is present in the epithelial cells. In the same way, the P N cadherin. N cadherin. Here is one is N cadherin. You have to note down this N cadherin. N cadherin is present in nerve. Present in nerve and muscles and in the lens cells of eyes. ध्यान दीजिएगा, नर्व में, मसल्स में एंड लेंस सेल्स, राइट, लेंस सेल्स ऑफ आइज. ठीक है. P cadherin is present in placenta and epidermis. So now, my dear students, I think that the classical cadherin has three parts: E cadherin, P cadherin, and N cadherin. E cadherin molecule is present in epithelial cells, and N cadherin molecule present in this is a E cadherin that is present in epithelial cells, and the N cadherin present in nerve and muscles, and your next is the P cadherin is present in placenta and epidermis, and N cadherin also present in the lens cells of eyes. You have to note down. So this N cadherin is also expressed in the fibroblast cells. Those cells which forms the fibroblast. Fibroblast means, if you talk about the collagen fibers. So because the collagen fibers are also formed by the chondroblast. So the N cadherin is also present in fibroblast cells. You have to note down to all these things. These are very important things. The N cadherin is present in fibroblast cell also. Okay. E cadherin is expressed. This E cadherin, which is present in epithelial cells, this also expressed in the part of brain. This also expressed in the part of brain. Basically, it expressed in the midbrain regions. Basically, it expressed in the midbrain regions. And the different variety of cadherins which are expressed in the brain region, such as the R cadherin and the cadherin 6. So these are the these are classical cadherins: the E cadherin, P cadherin, and your the N cadherin. These are the classical cadherins. Because why they are called classical cadherin? Because their homology, their sequence homology. इनका जो sequence homology होगा, इसमें similarity होगा. So therefore these are the classical cadherins. The homology sequence means their domains, उनके कैसे domains हैं, as well as their amino acid sequence. And इसके अलावा instead of these classical adherent molecules, here is one you are seeing here, the T cadherin. This is a non-classical cadherin. Here you are seeing here, this is a non-classical cadherin. Why it is called a non-classical cadherin? Because that is the diversity in sequence diversity, on the basis of sequence diversity. Classical cadherin means sequence diversity is more, and in non-classical cadherin, sequence diversity is exists. Sequence diversity is very less in case of your classical cadherins. That means the more homology, and in non-classical cadherins the sequence diversity is more. You remember in that way. So now in brain, approximately in one brain, you have to note down, approximately in your brain, more than 50 non-classical cadherins are expressed in your brain. More than 50 non-classical adherents. More than 50 non-classical adherents are expressed in our brain. More than 50 non-classical adherents are expressed in our brain, and such as the protochordherins, and desmogloins that are from the desmosome junctions, as well as you will see here is a T cadherin. The basic difference between the T cadherin is Here is no transmembrane domain. This is a basic, very important student, very important for this one. Here you will see in this case of T cadherin. This is a question for your competitive exams also. The T cadherin is, it has no transmembrane domain. It is just like as a extrinsic protein, peripheral protein. This T cadherin, right? It is a non-classical cadherin. In you will see here, this is a proto-cadherin. It is non-classical. It is also a non-classical cadherin, and this has a transmembrane domain. And the cadherin 23, that is CDS 23, this is also has, it has many domains or transmembrane domain. And you will see the desmosomal cadherin, this is desmocolin, it is also a transmembrane domain. And the protein kinase cadherin, rat protein kinase cadherins, it has a extracellular intracellular domain. And this is just like as a G protein couple receptor 7 pass transmembrane cadherin. This is a question because in these cadherins here is no receptors, this like structure. This is a alpha helical structure. You will see here, this is a 7 pass transmembrane cadherin. What is this? That is a flamingo is a bird also. So it is 7 serpentine, it has 7 serpentine receptors. You will see here, this is very important. ये पूछा जा सकता है आपसे कहीं पर भी, क्योंकि ये यहाँ पर डिफर है बिल्कुल. इट इज वेरी डिफरेंट फ्रॉम द अदर्स. So flamingo has 7 pass transmembrane cadherin molecules, and this is a fat-like cadherin. Then this is the T cadherin. So T cadherin. Here is the important question: is T cadherin has no transmembrane. T cadherin has no transmembrane. T cadherin has no transmembrane. T cadherin has no transmembrane segments, no transmembrane domain. Okay, right? T cadherin has no transmembrane domain, and it is this T cadherin is attached to the plasma membrane of nerves and muscles. This T cadherin is attached to the plasma membrane of nerve and muscles, and by here is the it is anchored by a glycophosphatidylate inositol. This is a glycophosphatidylate inositol. This is anchored in the plasma membrane of nerve or muscles by a GPI anchor, and that GPI anchor is you have note down glycophosphatidylate inositol. क्या करते हो? तोड़ दो उसको. Glycophosphatidylate inositol, right? And now the fat and this fat-like cadherin, and this is a flamingo cadherin. This is a fat-like cadherin, and this is a flamingo-like cadherin that is at first identified in the Drosophila. That at first identified in the genes. These are the product genes of Drosophila. Drosophila is a Diprotodon insect, and these are the epithelial growth and cell polarity products. These are the flamingo and fat. These classical and non-classical cadherin molecules. This classical and non-classical cadherin molecules forms a cadherin superfamily. This classical and non-classical cadherin proteins forms a cadherin superfamily. In case of humans, more than 180 members. In case of the humans, more than 180 members, more than 180 members of these cadherin superfamily proteins are present in humans. Now I will just tell you this slide, and after that we will conclude to this one today's lectures, but the next lecture is ready. Now student, you will see here, this is the E cadherin. It is found in many epithelial cells. Main location is, and the junction association is adherent junctions. This is your adherent junction. This is your adherent junctions. And this is death of the blastocyst stage. Embryo fail to undergo the compactation. If this molecule is dysfunctional, this molecule is not governing to its role. And the N cadherin, that is found in neurons, heart, skeletal muscles, lens and fibroblasts. That I explain everything in previous sections. Adherent junction, chemical synapse, and embryos from the heart defects. Embryo is die from heart defect and if n-cadherin molecule is mutated. क्वेश्चन ऐसे ही आएगा, n-cadherin molecule में मोटिवेट हो गया तो क्या हो जाएगा, एम्ब्रियो की डेथ हो जाएगी हार्ट डिफेक्ट्स में. ऐसे नाउ द p-cadherin, प्लेसेंटा, एपिडर्मिस, ब्रेस्ट एपिथेलियम, एडिरेंट जंक्शन, एबनॉर्मल मेमोरी ग्लैंड्स. यदि इसमें सपोज दैट म्यूटेशन हुआ तो व्हाट विल द रिजल्ट इन, मेमोरी ग्लैंड्स विल बिकम abnormal development. V-cadherin, endothelial cells. Endothelial cells, these are the cells of blood. Adherent junction, abnormal vascular development. Apoptosis of endothelial cells. Blood vessels की apoptosis success. These are the examples of your classical cadherins. These are your examples of non-classical cadherin. Desmocolin skin में पाए जाते हैं. Desmosome is the junction and blistering of the junction. Suppose that इसमें कोई प्रॉब्लम आ गई तो blistering, जैसे जलने के बाद फपोले पड़ जाते हैं, इस तरह की कंडीशन है. Desmoglein, it is a skin desmosome, blistering skin disease due to loss of keratinocyte cell adhesions. ठीक है, keratinocyte cell adhesion के कारण blistering हो जाएगी स्किन में, फपोले टाइप के. Desmoglein that is non functional या it becomes mutated. P-cadherin present in neurons, transmembrane protein नहीं होती है. Neurons, muscles and heart, right? None, कोई जंक्शन नहीं है. Unknown, function known नहीं. Cadherin 23, inner ear, हमारे कान के अंदर. Other epithelia, links between stereocilia in the sensory hair cells. In the sensory hair cells, ठीक है, जो sensory hair cells होते हैं because they are pressure detecting cells. Suppose that ये cadherin 23 में कोई डिफेक्ट आ गया है, कई क्वेश्चन बन सकते हैं यहाँ से, इसलिए आप बहुत ध्यान से पढ़िए इसको. Cadherin 23 में कोई प्रॉब्लम क्रिएट होगी तो डीफनेस हो जाएगी, सुनाई नहीं देगा. Fat in Drosophila, epithelial and central nervous system. Signal relaying junction, planar cell polarity, enlarged imaginal disc and tumor, disrupted planar cell polarity. जो इमेजिनल डिस्क होती है वो इनलार्ज हो जाती है, इसमें आ गया. ऐसे फैट वन इन मैमल्स, वेरियस एपिथेलिया इन सेंट्रल नर्वस सिस्टम. स्लिट डायफ्राम इन किडनी, किडनी में डायफ्राम स्लिट एंड ग्लोमेरुलर सेल जंक्शन. स्लिट डायफ्राम, माल फॉर्मेशन ऑफ फोर ब्रेन एंड आइज. ठीक है, फोर ब्रेन और आइज का डेवलपमेंट नहीं हो पाएगा. अल्फा बीटा एंड अल्फा बीटा गामा प्रोटोकैडरिन्स, दे आर प्रेजेंट इन न्यूरॉन्स and these are chemical synapse, non-synaptic membranes, neuronal degeneration. And flamingo, sensory and some other epithelia, cell junction, disrupted the polar and tube effects. So these are the this is the story of the cell communication part 1. If you have any questions regarding to this one, so you can ask to me. If you have any questions regarding to this one, हाँ ये very good, ये तो question हो गया इसका. If you have any questions regarding to this part 1 of the cell communication, because today I just tell you about the introductory part of this cell communication, then this is the connective tissue, extracellular matrix and cell to cell interaction. Then these are the 4 types of the junctions: anchoring junctions, occluding junction, channel forming junctions and signal relaying junctions. After that I just to explain to this this part. Occluding junction बता दो. Yes, I will tell you the occluding junctions. देखिए, occluding junctions is a tight junction. Number 1, occluding junction is a tight junction. Occlude junction is tight junction. Tight junction completely seal कर देगा ये tight junction. ठीक है, completely seal कर देगा. और vertebrates में tight junction होंगे और invertebrate में separate junction. I will show you. This is the occluding junction. ये occluding junction दो cells के बीच में completely separate कर देगा, जिस कारण से नो मूवमेंट ऑफ द मॉलिक्यूल्स फ्रॉम वन सेल टू द अनदर सेल. ओनली स्मॉल मॉलिक्यूल्स विल गो. बहुत छोटे छोटे मॉलिक्यूल जा पाएंगे, बड़े मॉलिक्यूल नहीं जाएंगे. तो या तो सेल इम्परमेबल हो जाएगी या सेल सेलेक्टिव परमेबल. हाँ, दूसरा क्वेश्चन, वेरी गुड मनीषा का क्वेश्चन. सर ड्रोसोफिला में कौन सी फर्स्ट आइडेंटिफाई हुई थी? फैट एंड फ्लेमिंगो. फैट एंड फ्लेमिंगो, आई थिंक दैट, हाँ, फैट एंड फ्लेमिंगो, ये दोनों जो हुई थी, आपका एक फैट और दूसरा आपका यहाँ पर ये जो 7 ट्रांसमेम्ब्रेन कैडहेरिन फ्लेमिंगो है, ये दोनों ड्रोसोफिला में रिपोर्ट हुई हैं. सबसे पहले दीज आर दी प्रोडक्ट ऑफ जीन प्रोडक्ट ऑफ ड्रोसोफिला न्यूरोगास्टर. देखिए इंपॉर्टेंट पॉइंट यहाँ पर ये है, पहले ये वाला है फैट कैडहेरिन. ये जो 7 पास ट्रांसमेम्ब्रेन कैडहेरिन है, 7 बार ये जो है कैडहेरिन मॉलिक्यूल यहाँ पर आपका सर्पेंटाइन रिसेप्टर, इज जस्ट लाइक ऐसे सर्पेंटाइन रिसेप्टर. आप कोरिलेट करिए टू द जी प्रोटीन कपल रिसेप्टर. ओके, योर क्वेश्चन इज ओके. नेक्स्ट